welcome to the video lecture on the environmental models of autism. So we previously talked about the different tests of ASD, the phenotypic models, and the genetic models. So this is the last of the models of autism. So here we're going to talk about prenatal valproic acid, neonatal Borna disease virus model, and the maternal immune activation, and how all of these models are used for environmental models of autism. So prenatal exposure to valproic acid, which is an anticonvulsant, is known for risk of development of ASD. So prenatal exposure in rodents produces behaviors that are consistent with ASD. So this is why in a lot of patients that have epilepsy are told when they're pregnant to actually stop taking their epileptic medication, because valproic acid can be in a lot of these anticonvulsants and can actually harm the fetus. So they will actually stop taking their medication to help prevent or lower the risk of their fetus developing certain developmental diseases such as ASD. And so in the model of ASD where we see the exposure to valproic acid, then some findings we see is a decreased social interaction. We see decreased sensitivity to pain, so an increased sensitivity to non-painful stimuli. We see repetitive stereotypic-like activity, increased anxiety, long-lasting fear memories, which are difficult to extinguish, and altered isolation-induced vocalizations. So you can see how a lot of these endophenotypes are very similar to the phenotypes of ASD patients. So in the VPA model, so VPA is just valproic acid, VPA. Then we see decreased spine density and increased dendrite length in these axons. So remember we talked about that in part 3 lecture. And so it's sensitive of hypo and hyperconnectivity phenotypes. We also see that some behavioral interventions can reverse the VPA phenotypes. So that would be environmental enrichment and multisensory stimulation can really help turn these around. So this environmental enrichment and multisensory stimulation show and ameliorate some symptoms in autistic children. In other words, these are treatments that we use in autistic children as well as in these animal models. So that would suggest perhaps there's some good predictive validity here. So now let's talk about a different model, the maternal infection. So maternal infection is associated with an increased risk of developing autism, as well as other disorders and diseases, so such as schizophrenia. We talked about that as well. And a study by the Danish Medical Register looked at over 10,000 cases of autism and looked at where, during what point of pregnancy the infection may be more dangerous at, or what type of infection is most dangerous. And it's really interesting to note that the strongest association they found was with a rubella infection. In other words, the strongest risk of developing autism was if the mom got infected with rubella during pregnancy, which is interesting since rubella, is preventable by the MMR vaccine. the exact same vaccine that our horrible friends, the anti-vaxxers, say cause autism. It's amazing how that vaccine can actually prevent autism. Weird. And so further evidence exists also using peripheral markers of inflammation with the strongest correlations between cytokine IL-6 and autistic behaviors. So the maternal immune status is also associated with ASD. So mothers of ASD children have elevated levels of autoimmune diseases, so including arthritis, celiac disease, and type 1 disease. There's also evidence of inflammation in ASD children that will persist into adulthood. So again, we see elevated cytokines and pathology of microgliosis and astrogliosis in the postmortem ASD brains. We also see elevated cytokines in the cerebrospinal fluid of ASD children and dysregulation of immune genes in the autistic brain was seen in a microarray studies. So an infection while pregnant, so as a fetus, when the mom's infected, it will not only affect her, but it will actually change the child itself. It'll change the immune system of the child well into their adulthood as well. So neonatal Borna disease is another disease that's kind of associated with ASD. So neonatal Borna disease virus infection. So this is a major human risk for ASD, would be rubella, is a human-specific infection. So Borna disease virus is a persistent neurotropic virus capable of infecting humans, rodents, horses, sheep, cattle, and birds. Causes persistent non-lethal infection of the neural cells. And it's analogous to an in utero rubella infection. So we use this model, the Borna disease model, to mimic an in utero rubella infection in rats. So the BDV model would be that rats are infected at birth, so neonatal period analogous to the third trimester in humans. And then we see some neuroanatomical deficits. So we see this in the cerebellum and the limbic system. So here's the cerebellum in the rat brain, and then here's part of the limbic system here. And then we see behavioral deficits such as hyperreactivity, some circadian rhythm disturbances, social play deficits, cognitive deficits, and chronic anxiety in the animal model. So again, there are of course limitations we need to talk about with this model. So the problem is that BDV is a persistent infection, so it's not an infection that gets better. Whereas the rubella infection does eventually get better. Also, that infection is in the mom, whereas this BDV infection is actually in the patient itself, or in the animal itself. And so this isn't seen in ASD. Also, we need to talk about the timing of insult. So the neonatal third trimester does not match the risk of the viral infection in ASD. And it may actually reflect the specific neurotoxin effects of an infection rather than the maternal immune activation. So again, we're infecting the patient, not the mom. So it's largely supplanted by MIA models for ASD. So, this is a decent model, but we don't use it as much because we tend to use models that are actual maternal immune activation models for ASD. So this would be the polio C model is usually the preferred system. So let's talk about that one. The polio C effects are moderated indirectly through placental transmission of inflammation. So this is the key role for IL-6 in placental transmission of inflammation. So in this image here, then you can see that. So we've got the maternal immune activation leads to our polio C, the pro-inflammatory C cytokines. That's polio C. I don't know if I said that. And what that does is that comes down in here. So we see the immune cell activation, and what we get is that IL-6 from the maternal bloodstream comes in along with the IA-induced placental IL-6 and comes here. So then we get activation of this, which you don't need to know, and it leads to downstream effects on fetal development. So you can see how this is in the placenta, and then it leads to the fetus. Okay, so that's how MIA can lead to changes in the fetus itself. So this is really the number one model, is to infect the mom somehow, which will then lead to these IL-6 from the maternal bloodstream, and using this as placental transmission of inflammation, which is then transmitted down to the fetus itself. So while the BDV model is good, you need to remember that it is not a replacement for the maternal immune activation, because in that then you're activating the immune system of the patient itself and not the mother. So that is it for our models of research models of autism, and that's it for all the lectures on autism.